What did the very first proteins look like, those that appeared on Earth around 3.7 billion years ago? Professor Dan Tawfik of the Weizmann Institute of Science and Professor Norman Metanis of the Hebrew University of Jerusalem have reconstructed protein sequences that may well resemble those ancestors of modern proteins, and their research suggests a way that these primitive proteins could have progressed. Their findings were published in the Proceedings of the National Academy of Sciences, PNAS. The proteins encoded in a cell's genetic material are the screws, springs and cogs of a living cell, all of its moving parts. But the first proteins, we assume, appeared well before cells and thus life as we know it. Modern proteins are made of 20 different amino acids, all of them essential to protein building, and all arranged in the form of a polymer, a long, chain-like molecule, in which the placement of each amino acid is crucial to the protein's function. But there is a paradox in thinking about how the earliest proteins arose. Because the amino acids needed to make proteins are themselves produced by other proteins, enzymes. It's a chicken and egg kind of question, and it has only been partially answered until now. Scientists believe that the very first true proteins materialized from shorter protein segments called peptides. The spontaneous generation of amino acids had already been demonstrated in 1952, in the famous experiment by Miller and Urey, in which they replicated the conditions thought to exist on Earth prior to life and added energy like that which could come from lightning or volcanoes. Showing amino acids could, under the right conditions, form without help from enzymes or any other mechanism in a living organism suggested that amino acids were the egg that preceded the enzyme chicken. Tawfik, who is in the Institute's Biomolecular Sciences department, says that is all well. RNA is today presumed to be the original molecule that could both carry information and make copies of itself. So contact with positively charged amino acids would theoretically be necessary for further steps in the development of living cells to occur. But there was one positively charged amino acid that appeared in the Miller-Urey experiments, an amino acid called ornithine that is today found as an intermediate step in arginine production, but is not, itself, used to build proteins. The research team asked, what if ornithine was the missing amino acid in those ancestral proteins? They designed an original experiment to test this hypothesis. The scientists began with a relatively simple protein from a family that binds to DNA and RNA, applying phylogenetic methods to infer the sequence of the ancestral protein. This protein would have been rich in positive charges, 14 of the 64 amino acids being either arginine or lysine. Next, they created synthetic proteins in which ornithine replaced these as the positive charge carrier. The ornithine-based proteins bound to DNA, but weakly. In Metanis' lab, however, the researchers found that simple chemical reactions could convert ornithine to arginine. And these chemical reactions occurred under those conditions assumed to have prevailed on Earth at the time the first proteins would have appeared. The scientists also discovered that in the presence of RNA, that the ancient form of the peptide engaged in phase separation, like oil drops in water, a step that can then lead to self-assembly and departmentalization. And this, says Tawfik, suggests that such proteins, together with RNA, go. Professor Dan Tawfik is the incumbent of the Nella and Leon Benezio Professorial Chair. Story source, materials provided by Weizmann Institute of Science. Note, content may be edited for style and length. Thanks for watching. Do you like my video? Then show me your love by pressing like button. Please subscribe my channel and hit bell icon for new science inventions in future.